Okay guys, let's get back to video number 2 for Unit 9 now. We just talked about the formation of plaque and how microorganisms are involved in that. So what we're going to talk about now for the rest of this unit is how these formations of plaque or bacteria can go from something as simple as, a biofilm on our teeth to something as serious as heart disease and stroke. So we look at the steps here on this slide. It's a pretty good organization of it. When we get our bacteria, it grows in our mouth because it's got lots of food, it's got moisture, it's got sugars that they use to grow, and if we don't take care of our mouth, we have poor hygiene, then we get the formation of plaque. So we talked about that in the last video, and we know how that forms. We know a little bit about it now. So where does plaque go once it's formed? Well, one of the things that happens is it helps bacteria stick to your teeth. It's a biofilm. Those bacteria are going to produce waste products, and just like we talked about when we talked about bacterial metabolism, one of the things that bacteria can do when they ferment is they produce acids. Now, if they produce acids and they're stuck to our teeth, then this acid can dissolve the enamel that's on the outer part of our teeth. This can lead to, of course, a cavity or a carie. So that's a hole in the enamel, and if you go to the dentist and you have a cavity, then the dentist needs to do an intervention in order to fix it. So that's one thing that can happen. Another thing that can happen is these acids can also lead to gum disease. The irritation that you get around your teeth and your gums turn really dark and they get swollen and they can bleed when you brush your teeth. Well, that's a condition called gingivitis. Gingiva, which refers to the gum tissue, and itis generally means to a disease or infection. So plaque so far is going to go on. It could cause cavities, and now it could cause gingivitis. Now, as this plaque hardens and it forms into tartar or calculus, that we talked about a bit earlier, this tartar and calculus can move deeper down into the tissue between the gums and the teeth, and this can attack the tissues that are in our periodontum, and this can lead to a disease called periodontitis. And we'll talk about that later on in our unit, and that's a disease that attacks the bones and the ligaments of the tooth and the mouth. And that's quite a serious condition when we get to that far, and generally we need some help from our dentist when that happens. So on this slide here, we're looking at gingivitis. We talked about in the last slide, which is the inflammation of the gums. And if you look up here, you can notice there's some red and some redness next to the gums here by the teeth. That's a sign of gingivitis. We can treat this because it's easier to treat, because it's only affecting the gums. So if we get rid of the plaque and we get rid of the bacteria there, the gums will heal themselves and we'll go back to a healthy condition. If we left this untreated, what's going to happen is these infections here will move further into the underlying tissue. And then that's when we get into periodontitis. Now, on here, you can see the gingiva is not quite as healthy anymore. You can see how it's receded. It's gone down on this tooth here and on this one here as well. So we've lost gingiva. So we're getting into early stages of periodontitis, and if we get into late stages, now we're getting damage done to the base of the tooth, the root of the tooth, and there's probably going to be damage done as well as the bone that comes in between these teeth right here. Not only does it could cause tooth loss, you could have to have surgery, but it could also lead to more serious conditions like heart disease, stroke, or diabetes, and we'll talk about that later in this unit. Okay, so we talked about plaque, we talked a little bit about how it forms, so now let's talk about biofilm formation or biofilm morphogenesis. Morphogenesis is a big word to mean, morph means changing and genesis means to create. Well, basically morphogenesis means a change in shape. Or a change in the way it develops or grows. And there's 3 or 4 different steps. We've got 5 listed right here about how we get a biofilm or plaque formation. So we start with a pellicle, then we go to bacterial adhesion, then bacterial colonization, plaque growth and maturation, and finally biofilm and matrix formation. So we're going to talk about that now over the next few slides, and hopefully we'll give you a good picture about how this comes together. So this picture here, it's pretty good, but it goes through this couple of steps that we're talking about. So the first couple of steps we get here are the pellicle formation. So you can see the pellicle down here. So here's our enamel from our tooth, and then the pellicle forms right on top of our enamel. And then that pellicle is a thin layer, and we'll talk more about it in a second, but it allows bacteria to adhere, to stick, and then it allows others to colonize. So once we get a layer of bacteria that sticks on top of that pellicle, then we get other bacteria that can move in and on top of it and form layers. Now, as this biofilm grows in height, it'll start to release cells to move away. So these cells here now, if they move from here, they can get released. They can move to other parts of pellicles, and they're already going to be able to start forming new biofilms. So this is how a biofilm would extend and how it would grow out. Now, I don't necessarily want you to worry about all of the names of the steps in this picture here, but what I do want you to know is if you look at the complexity of growth. So if we look up here in step A, you'll see we're talking about associations. This is basically a formation of our pellicle. And you can see that here we've got just a little formation of the pellicle on top of the enamel. So these blocks are going to represent the surface of the tooth, the enamel, and this is going to be the formation of the pellicle on top of it. And you can see there's, now that we get the formation of a pellicle, we can have some bacteria move in and they can stick to there. So this is going to be our attachment or adhesion to the pellicle. And once the bacteria get there, of course, one of the things they're great at is being able to grow and reproduce. So we're going to have, multiplication and colonization, and then these cells will be able to branch off, and those cells branch off and they'll be able to move down into other parts of the tooth, other places where they can find pellicle, and then they can add to the biofilm. Finally, we get down to these stages where we get the maturation of the plaque. The plaque's getting deeper. We're getting more and more layers of bacteria here. You can see the dark pink are going to be our matrix. And then finally we get down into maturation, and maturation is a step where we've got full-on dental plaque formation. This is definitely going to have to be removed by a dentist for now. And there's even channels that run through the matrix that allow waste of the bacteria to get out and fresh nutrients and water and air to get in. So it's a really complicated structure, and this is a good picture of how it all starts. We start quite simple with a pellicle. We move into colonization and adhesion. Then we get the bacteria moving around, being able to move and go to other places. And then we get the maturation of the matrix. And finally we get the full-on formation of plaque. So this pellicle I've been talking about, this is basically step one when we're talking about plaque morphogenesis. The minute the dentist stops cleaning your teeth, we start to lay down a pellicle. It's something we can't stop, sadly, but it's the way it goes. And the thin pellicle is just a layer of proteins, glycoproteins, sticky proteins that are present in our saliva. So as soon as fresh saliva touches our teeth, then we get a layer of pellicle. This allows the bacteria to attach to the pellicle, not the enamel. It'd be much tougher for bacteria to stick to the enamel because it's a very smooth compound. So in day one, we've got the formation of the pellicle. In day 1 and 2, now bacteria are going to stick to that pellicle, and they're going to migrate through the pellicle towards the enamel of the tooth. So another word for enamel is hydroxyapatite, but I'll use enamel. They're going to be adhesions on these different bacterial cells that we talked about earlier, and those adhesions are going to allow other bacteria to stick to them. So the bacteria pili and fimbriae, these will attach to the pellicle, and these bacteria will move closer into the enamel. And we get a bunch of gram-positive bacteria at this point, and we call them colonizer or early or pioneer bacteria. And these are generally of the Streptococcus genera. I'm not expecting you guys to remember the names to these species, but there are some important ones that we'll talk about later. Now, this is going to very similar to the slide I showed you a few minutes ago, but it's going to show you a little bit more detail. And then we've got our tooth surface, we've got our acquired pellicle, and then we get our colonizer bacteria that move in on top of it. Once we get these colonizer bacteria, then we get these adhesins, and then all of this other bacteria is much more easy for them to come along. So we get our early colonizers, and then we get our late colonizers that come later in the process. So when we get to day 2 or 4, our pioneer species have attached to the pellicle, and they've laid down, these organic products, these waste materials, and that's going to help other bacteria species survive. So when they get there, there's places for them to grow, there's stuff for them to eat, and so on and so forth. So the early colonizers are going to be our gram-negative cocci and rods, and they're going to probably be facultative anaerobes, so they can live in both aerobic and anaerobic conditions. So these are going to happily attach to those bacteria, those streptococcus that came down first and provided that first layer for bacteria to grow on top of. This layered system is going to keep happening. So we're going to have layers of bacteria coming in. They're going to lay down their metabolic products, like these insoluble sugars, these extracellular polysaccharides, and that's going to allow other species of bacteria to come in on top of it. So these sugars and extracellular polysaccharides are basically just going to be a sticky substance that bacteria can stick to. It's going to create this biofilm, and then as a result of that, these bacteria below the biofilm, they're going to be sheltered from the immune system. So they can basically grow without any worries of a predator, which is our immune system, coming in and disturbing it, or disturbing them, sorry. Now, if we move on to day 4 and 7, so by the end of the first week, we've got our late colonizers here already. Now we're going to have gram-negative cocci and rods. These next ones that are going to move in are going to be strict anaerobes. They're going to be anaerobic. They won't be aerobic bacteria whatsoever. And you'll see different shapes, the filamentous, the fusobacteria. We'll see spirochetes and spirals. So by week 7, we've gone from just streptococcus aerobes to strict anaerobes. Now we're talking about rods, we're talking about spirals, we're talking about spirochetes, filamentous bacteria. So now we've got a very large diversity of species living in this biofilm. Now, after the first week, we've got the formation. Now we've moved into the growth and maturation stage. So this is talking about the second week of maturation for these biofilms. We've got these anaerobic bacteria. They're going to start to cause inflammation of the gingiva. So we're getting into very early stages of gingivitis at this point if the biofilm is left completely intact and it just does its thing without any external pressures like brushing or dental intervention or anything like that. We'll actually get to the point where white blood cells are going to appear in the plaque to try and stop these bacteria from growing as best they can. And after the second week, we'll start to get clinical gingivitis, which is we're going to see symptoms of gingivitis. We're going to see red gums. They're going to be inflamed. They're going to be tender. They may bleed if you brush or floss. That's clinical gingivitis. We've damaged now not only perhaps the tooth enamel, but we've also got deeper and we've damaged the gingiva. Again, we're going to see an increase in the diversity of species. And what we're going to see now is the matrix is going to form itself into these mushroom shapes, these shapes that rise up off the teeth enamel. And at the bottom of these, we're going to actually find these channels. So these are going to be spaces, empty spaces. And what this is places for bacteria waste to be excreted so that it gets away from the cells. And it's also going to carry fresh nutrients or oxygen if the bacteria needs oxygen, so that this biofilm the plaque gets to grow. And this is going to start to become very hard, and that's when we get into the substance called calculus. So here we start off with these pioneer species. We're healthy. You can see how the diversity increases in the biofilm, and eventually we've got a much thicker biofilm. We penetrate it into the periodontum, and we've caused periodontal disease. So we go from a healthy tissue to a diseased tissue in about 3 weeks if everything's left untreated. And here's a look at the complexity.